Welcome to Dysautonomia International's May 2021 webinar. My name is Lauren Stiles, and I am the president and co-founder of Dysautonomia International. I'm very happy to be joined this evening by Dr. Todd Levine, who is a fantastic neurologist and neuropathologist in Arizona with Honor Health. And he has a really exciting talk for us on a new topic that we know a lot of patients are very interested in. He completed his residency in neurology and a fellowship in neuromuscular disease at Washington University School of Medicine in St. Louis, Missouri. He's been a neuromuscular specialist for over 20 years, and he's currently the director of the neurology department at Honor Health in Phoenix, Arizona. He is also board certified by the American Board of Psychiatry and Neurology and the American Board of Electrodiagnostic Medicine. And I think Dr. Levine has a ton of credentials, but I know everybody wants to get to the talk and hear the topic. I just want to remind everyone that you can ask questions during the talk. If you scroll down to the bottom of your Zoom screen, there's a Q&A button. So you can ask a question. Usually what happens during our webinars is that we get way more questions than we could possibly respond to in the time allotted. So if your question isn't answered, please don't be mad at us. It's just a matter of having the time to get through everything. Generally speaking, we ask that you don't ask personal medical questions. Dr. Levine can't give individualized medical advice this evening. And the more generally applicable your question is, the more likely it is we'll be able to ask that question. I also wanted to thank our partners who help us make these webinars free throughout the year. Tonight's webinar is co-sponsored by SaltStick, Liquid IV, and Normalite, and they're all great electrolyte hydration products who help our patients out. So we thank them for their support. And with that, I want to turn it over to Dr. Levine to teach us about this exciting new topic.
Great. Well, thanks, Lauren. Yeah, so I think some of you may have seen that last month we wrote a paper on this topic. And the idea behind the paper really was to see what we could do to kind of further the understanding of the specific pathology that's present in people with POTS. So I will try to take you through that, see if I can advance my slide. Yep. So just a few disclosures. I do work with and own parts of two individual pathology labs. One is called Corinthian Reference Labs, that does small fiber testing, and then the other is called C&D Life Sciences that does the testing that we're going to talk about in this talk, which is looking for the protein called synuclein. I do some consulting for pharmaceutical companies, and then I have research support, as you see here. So I want to kind of start off with some basic premises that I think are really important. And the whole basis of science is the idea of formulating a hypothesis, a theory, and then testing that theory. And the best scientists throughout history are the ones that will do that, but they'll not be kind of glued to their hypothesis. So they're just as likely to disprove their hypothesis as they are to prove it. So Enrico Fermi, who was the architect of the nuclear age, won the Nobel Prize for developing nuclear fission, wrote that there are two possible outcomes. If the result confirms the hypothesis, then all you've done is made a measurement. But if the result is contrary to the hypothesis, then you've made a discovery. And so the idea that he, this was brilliant physicist, would make these hypotheses and be happier when he did the science that disproved his hypothesis, I thought really kind of spoke to his brilliance. And then Robert Pirsig, who many of you may have read the book Zen and the Art of Motorcycle Maintenance, he wrote, for every fact, there's an infinity of hypotheses. And I think that POTS is a great example of this. We know POTS is a fact, and we have many, many hypotheses as to what causes, how to treat and cure this disease. And then this is me. So my quote was that the best thing I do as a neurologist is telling patients I don't know. And I say that a lot, which is true because in neurology, we lack tests for so many things. The reason many medical students and many medical students don't go into neurology is because there are no easy answers. There's no definitive answers. You can't just say someone has a glucose of 300, therefore they have diabetes. Neurology is the art and the science of taking all of the facts that one has available and making the best possible hypothesis. But like Fermi, the best neurologists are the ones that can recognize what they don't know, because trying to recognize what you do know when we don't know the facts is what gets us into trouble. So I think this talk, if you keep those things in mind, will be very important as you sort of look through, I guess, my mind, if you will, how I try to understand these conditions. So as we think about POTS, one of the problems, as I said, is that we have not had traditionally a great diagnostic set of criteria. We have not had a clear consensus on what this is. And so if you look over the history of this syndrome, what you find is that it's been called many, many things. You know, if someone needs 15 different descriptions of a clinical syndrome, that probably means we don't have the one good clinical description of that syndrome. And I think this has, again, been one of the problems in trying to understand POTS. So there has been a consensus statement that came out about 20 years ago. And this was the clinical definition. So what they said is that when you stand up, when you have a postural increase in your heart rate of 30 beats or more without dropping in the blood pressure. So if the blood pressure drops and the heart rate goes up, that's really indicative of something called neurogenic orthostatic hypotension. What we're talking about with POTS is the heart rate increasing, but the blood pressure is staying stable. You can also see people that just simply have a standing heart rate of 120 beats per minute, which is consistent. And then in children, we actually want to see more than 40 beats per minute increase to be consistent with the diagnosis of POTS. As that heart rate increases, there is what we call orthostatic intolerance. And so that is the development of the symptoms that occur when you stand up that then resolve when the patient lays down. This really is sort of the best clinical description that we have for POTS at the moment. So why does this happen? Well, it happens because when you stand up, all the blood is in your bloodstream. And in the normal person, as we stand up, there's about 1 liter of blood that's going to get shifted from the central part of the vascular system to the peripheral part of the vascular system. The heart senses that. The heart senses that the blood has kind of left the central part of their body. And so it will increase the heart rate to try to keep pumping the blood and to maintain a consistent blood pressure. So that's what occurs sort of in the normal person. And then within about 30 seconds, there's a new baseline. So there's a very quick increase in heart rate, and then it very quickly comes back to the new baseline. And so this is what you would see in a normal person. Their resting heart rate is about 60. As they stand up, it may go up to 80 or 90, but within 30 seconds, it has come back down to maybe 70. So almost back to the baseline because they're still standing. So this is a healthy response to the postural change in position. If you have POTS or orthostatic intolerance, as you can see in this diagram, what happens now is when you stand up, the heart rate goes from 80 or 90 up to 140, and it stays that high for 10 minutes or more. And so this is a very abnormal response to the postural change. And with that come the symptoms that you all and I am very familiar with. So patients have lightheadedness, their vision can blur, they will have palpitations, they can feel their heart rate increasing, they can feel weak, fatigued, they will start to feel short of breath, they have anxiety, chest pain, nausea. all of the symptoms that are associated with POTS, which are due to this orthostatic intolerance, their heart rate having to work more to be able to maintain that normal blood pressure. So the question then with POTS is, does everyone who has those symptoms and that change in heart rate associated with the no change in their blood pressure, is that all one group of people, or are there individual groups? And so in medicine, we often talk about people being lumpers, meaning, well, just take all POTS, put them all together, that's going to be one group. Or we talk about them as being splitters, which is, can we break them into little groups? The advantage of breaking people into little groups is that you may find that one group has a specific pathology, or one group responds better to a specific type of treatment. Whereas if you lump them all together, it's going to get kind of washed out and you're not going to get to see those differences. So I trained with a really brilliant neurologist at Washington University who pre-computer and pre-internet everywhere would carry around little, spiral binders. And when he saw patients, he would write, this person had POTS and wore a blue shirt. And then he'd look for every other person that might have had POTS that wore a blue shirt, and he'd put them together and he'd think about them as a group. And he would say, does this make sense? Is there a reason to think that these people all come together and are more consistent than the other people with POTS, for example? And he really, I think has changed much, not about POTS or dysautonomia, but really has changed much about the way we think about neuropathies in general, because he was such a good splitter. Because of that, I've always sort of had that kind of inherent bent, that when I see people, I'm not just thinking about, okay, what's their diagnosis, but is that person with that diagnosis like everybody else with that diagnosis? And I think if you think about it, what you'll see is that in POTS in particular, there are very different types of people with POTS. Now, that may be telling us nothing, right? And my hypothesis is that that tells us something, and it may not. I could be wrong. Or putting people into smaller groups might be helpful in terms of how we think about them. So two groups that we often think about in talking about POTS, one are called hyperadrenergic and one is called neuropathic. So what are those two groups? Well, hyperadrenergic POTS are patients that when you do the tilt, when you stand them up, their heart rate increases by more than the 30 beats per minute. But in addition, their blood pressure goes up. And again, remember, in most POTS patients, their heart rate will go up and their blood pressure will, in essence, stay the same. But we do see this group of people that have a blood pressure that goes up. And that may have to do with increased catecholamine levels that are circulating in response to the tilt, or just in general in those people. The other group of patients that may be kind of split off, we now call neuropathic POTS. And the way I sort of think about patients with neuropathic POTS is that these are patients where we have objective evidence that there's something wrong with their nerves. Now, it doesn't mean that the other POTS patients don't have that. It just means that we haven't been smart enough to figure out how to identify them. So how do we identify patients with neuropathic POTS? Well, one would be autonomic testing, like the QSART, looking at the pseudomotor axonal reflex test or their sweat response. If that's abnormal, that is an objective piece of evidence that says that their sympathetic nerves involved in sweating are not functioning correctly. That would then lead us to say that's a neuropathic group. The second would be that when we can do skin biopsies, we can look at these small unmyelinated nerves in the epidermis. And these small unmyelinated nerves are also involved in the autonomic system. So finding evidence that there's damage to those nerves, again, tells us this patient we could lump as neuropathic POTS. And then the question is, are there other ways to identify that there's a problem that we can see in patients' nerves? There certainly are imaging techniques and other modalities, and even using spinal fluid. So there are many ways to try to show that this patient clearly has some damage to their autonomic nervous system. These two groups don't have to be exclusionary. So you can have a patient that has neuropathic POTS who also has hyperadrenergic POTS, or they can be separate. But it does allow us to think about these patients just a little bit differently and to start to put them into these smaller buckets. So what do we know about neuropathic POTS? Well, again, what we know is that this group of patients has some evidence that we can see visually, pathologically, that there is damage to the small nerve fibers and or the autonomic nerve fibers by doing just a simple skin biopsy. So taking this little piece of skin, and this is sort of an example to show you. So this is a quarter. This is the size of the piece of skin that we have to take in order to do this test. So it's very, very small. It doesn't involve any stitches, very simple procedure to do. But we can then stain these nerves. So these individual fibers, these little black squiggly lines, are coming up to the surface of the skin. So when you touch your skin and you can feel that, what is sending those signals for that sensation are these little fibers that we can now visualize. So these are the nerve fibers involved in the sensory nerves. They're in the epidermis, in the very outermost layer of the skin. I'll show you in a second. We can also look a little deeper than that. We can look into the dermis, which is where the autonomic nerves are, and we can study those as well. And so in patients that have a small fiber neuropathy, what we do is we count the number of nerves. So on the top here, you'd see the number of nerves in a normal person. And then down here, you can see this long gap with no nerves because the nerves have died off. And as those nerves die, we talk about an epidermal nerve fiber density, or the number of nerves per millimeter of skin. When that number is too low, that's objective evidence that there is a neuropathy that's present. And taken together with a neuropathy, patients with symptoms of POTS, that's how we define neuropathic POTS. Okay, so most of you have probably studied this already, so I won't go into too much depth, but the question is, what do we know about what causes POTS? And we know of a lot of things that cause POTS. So POTS is not that rare a condition. Depending on what you read, it's probably somewhere between 1 and 3 million people in the US. So it's a relatively common condition. So you can get there by having not enough blood volume. So patients that are dehydrated or have other medical conditions that cause them not to be able to resorb or absorb their fluids, or they're dumping fluids, can cause hypovolemia, and that will cause POTS. I told you that when we stand up, the blood leaves the heart and goes out into the periphery. So some patients will actually have abnormal venous pooling. These, not always, but these patients will say that their feet can turn red or purple when they stand up very, very quickly. And that's because there's too much blood in their legs. And if there's too much blood in their legs, there's not enough blood in their heart, and it's going to lead to POTS. Deconditioning is definitely a cause of POTS. Probably one of the best examples of this, and I think one of the things that kind of spurred a lot of attention in POTS in the 80s and 90s was actually in the astronauts. So the astronauts were perfect physical specimens. They'd go up into space, spend a year in space, come back, and every time they stand up, their heart rate would race or they'd even pass out. And that was because their heart didn't have to work against gravity for months and months at a time. And so you just had to recondition their heart to do that. More serious conditions. So one is that you could, again, have problems with your autonomic nervous system, so that the signals to the heart and the signals to the blood vessels that are supposed to be regulating the heart rate and the blood pressure are not doing what they're supposed to do. That can occur either because there's central dysregulation, meaning the brain and the spinal cord, or it can occur because there's damage to the nerves out in the body itself, which is, again, what we call neuropathic POTS. We know of a variety of different autoimmune conditions that can lead to POTS, as well as leading to these types of neuropathies, such as the acute autonomic ganglionopathies can do this. Some of the newer antibodies like TSHDS and FGFR3 are getting published on as causing this, Sjogren's syndrome, lupus, sarcoid, many autoimmune conditions that can lead to POTS. And then we know of genetic disorders. So there are rare genetic disorders that probably cause POTS primarily. And then there are genetic conditions like Ehlers-Danlos syndrome that can lead to POTS as well. So a big variety of diseases that can cause this condition. So how do we think about this? And I just want to kind of review how I think and how neurologists think in general. So if you come into a neurologist to see that doctor and you tell the doctor the symptoms that you're having, the first thing that the doctor has to do is to localize that problem. In other words, what part of the nervous system is not working? So for example, if I have numbness in my foot, that could be because of nerves in my foot or nerves in my hip or nerves in my back or nerves in my brain. What causes a brain problem is very different than what causes a foot problem. So I have to first, as a neurologist, know, well, am I thinking about a nerve problem in the foot, or am I thinking about a nerve problem in the brain? Once you localize the problem, then you try to identify the cause. Now, in neurology, we are pretty stupid. So we are not very good at this, and particularly when it comes to neuropathy. So for autonomic neuropathies, for sensory neuropathies, we probably identify the cause about 50% of the time. And I like to tell my patients, if I tried to get into med school with grades of 50%, I never would have gotten into med school. But now I live with that, right? I live with the fact that 50% of my patients, I can't figure out what causes the problem. And therefore, all I can do is to treat the underlying cause, okay? Or treat the symptoms. Those are my two options. If we don't know the cause of something, then we call it idiopathic, which means we don't know the cause. So despite lots of investigations into the causes of POTS, as well as autonomic testing, serologic tests, genetic tests, there's very unlikely to be a single unifying cause for POTS. There are going to be many, many causes for POTS, just like, again, there are many causes for peripheral neuropathies. And so we have to think in terms of how do we start to identify what those potential causes are. So that led us to what I did in this paper. And so the hypothesis in the paper is that there would be some patients with POTS who we looked for the easily identifiable explanation. So they didn't have hypovolemia, they didn't have deconditioning, they didn't have an autoimmune disease. And we still wanted to find, so that's the 50% of patients that we don't know the cause for, we still were trying to see if we could find what the cause might be. And the question then became, is there any reason to believe that these patients that have the, don't have other specific cause might be related to a more widespread autonomic disorder than POTS? So I run a pretty busy practice in terms of seeing autonomic patients and POTS patients. And so we reviewed retrospectively the medical history, physical exam, autonomic testing, and then the skin biopsies of the patients who were under age 50 who presented with a diagnosis of POTS to our service. The patients had all had symptoms that we would have labeled as being consistent with neuropathic POTS. So that was the group that we were interested in, because again, we were going to look at the pathology of these patients. So in total, we looked at 296 patients. Now, of those 296 patients, we excluded 274. And the reason we did that, again, is that this was a retrospective study. So this was not intended to be a prospective, all-encompassing study of the 296 patients. That would have been a wonderful study to do, but honestly, we didn't have the money to be able to do that. And then we said, of the patients that we considered to have neuropathic POTS, how many of those underwent a skin biopsy to confirm the diagnosis of neuropathic POTS? And the answer to that was 22 patients. So we're going to now focus on those 22 patients. And of those 22 patients, 7 of the 22 patients, we found abnormal phosphorylated synuclein in their cutaneous nerves. So again, I talked about the nerves in the dermis, not the epidermis. So the nerves in the dermis, which are all, or not all, but are primarily related to the autonomic nervous system. And we're going to talk a bit about what this means, but we looked for the presence of this particular protein. And so that broke into 2 groups. 7 of the 22 had the protein, 15 did not. And then we said, were there any differences clinically between the 7 and the 15? And so the answers were that those that had the phosphorylated synuclein, or we call that PSYN, were more likely to be male, were more likely to have some features of a sleep disorder called REM sleep behavior disorder. They reported less sleepiness and cognitive impairment than the 15 that did not. They had more GI symptoms, particularly gastroparesis. And interestingly, they were more likely to have that hypertensive response to the tilt table and abnormal QSART responses than the group that did not. And so what that sort of told us was, this is identifying a subset of patients that clearly are exhibiting more signs consistent with neuropathic POTS. So what does this protein do? What are we talking about? So let's talk about this a little bit. So these are from the paper. And if you look at D, E, and F, you see this bright green. And each of those bright green lines is an individual axon or an individual nerve in the dermis. And then if you look above that, this is the phosphorylated synuclein stain. So where you see the bright red, like here, through here, through here, that's where the phosphorylated synuclein is contained within those nerves. And the way that we know that is that we can use something called confocal microscopy, which will merge these two together. And so wherever you see orange in these pictures is the phosphorylated synuclein contained within an individual axon. And again, this was seen in the 7 patients that had the neuropathic POTS. So what is this protein and what are we talking about? So it's become clear over the last 50 years or so that a number of neurodegenerative diseases are due to a misfolding of proteins. So the most widely known is Alzheimer's disease. So this is a brain of a person with Alzheimer's disease. You can see this brown blob of protein here, and that's amyloid. And then also in Alzheimer's disease, you see these brown sort of extensions of these dendrites, and that's tau. So we've known that if you look inside the brains of people with Alzheimer's disease, you see this increase of amyloid and tau. Now, in Parkinson's disease, they have similar kinds of inclusions within their nerves, but they contain a protein called synuclein. And there are other neurodegenerative diseases that contain other proteins. So this has become a very common unifying theory behind these neurodegenerative diseases. And when we say neurodegenerative, what we mean is that the nerves over time will die and continue to die, so that people with Alzheimer's disease lose bits of brain and they continue to lose it and their memory gets worse and worse. People with Parkinson's disease have trouble moving and it gets worse and worse with time. This process of protein misfolding is kind of the single unifying theory behind all of these diseases right now. And there is a massive amount of study and work being done to try to figure out how to reverse this. So there is a protein in our nerves called synuclein. Synuclein is present in every nerve in our body. If you eliminate synuclein, say from animals, the animals will never develop. Their nervous system can't develop. We don't know yet what the function of synuclein is, but based on where it's found inside the nerves, we think that synuclein is very important in terms of releasing neurotransmitters from the nerves. So if one nerve wants to talk to another nerve, one of the main ways they do that is by releasing little chemicals. So norepinephrine, dopamine, all those chemicals. We think that synuclein somehow helps the release of those chemicals. I think I have, no, okay. So this is sort of the sequence of the protein. At the end of this protein, there are groups that can be added onto the protein. And when you add on this phosphorylation, this phosphorus group at the end of the protein, it causes the protein to fold incorrectly. So our schematic is, this is the normal alpha-synuclein. If that normal alpha-synuclein folds correctly, then it can go and do the biologic function that it was meant to do. On the other hand, if that protein folds incorrectly, it is now going to become toxic. And it's going to cause all of the other synuclein proteins in that nerve to misfold and build up. Then eventually it builds up so much inside of that nerve, it kills that nerve. And all those badly folded proteins leak out. And then they're taken up by nearby nerves and they start the whole process over and over again. And this concept of protein misfolding is now clearly behind a group of diseases that we call the synucleinopathies. Parkinson's disease is the most common synucleinopathy, but another disease called pure autonomic failure is also a synucleinopathy. So what we found then was that about 70%, or sorry, we found then was a group of POTS patients. We would say that about 70% of POTS patients will stay about the same. They don't have a degenerative disease. We haven't seen patients over the last few decades who get POTS just get worse and worse and worse and worse. It may happen to some, but about 70% that doesn't happen to. If they're treated, if they're managed, they can manage, they can get better. About 30% of patients may have a more progressive syndrome. We also know that about 30 to 50% of patients with POTS have neurogenic POTS. Now, we don't know that that's the same group. So not everyone who, say, has a progressive course necessarily has neurogenic and vice versa. But there is this group that we call neurogenic POTS. And then within that group, we now know that some of those patients may contain the phosphorylated synuclein. And containing that phosphorylated synuclein may indicate that they have early signs of a degenerative disease that is going to get worse. Now, what percent contain that protein is very difficult to answer. There's two ways to look at it from the paper that we did. One is we only found it in 7 patients out of the 296, because that's all that we found. The limit to that is that we did not test the 296. We only tested 22. And we also only tested ones that we felt confident had neurogenic POTS. So of the whole POTS population, it could be as little as 3%, but it could be as much as 30%. And until we do a larger, more prospective study, we're not going to have the specific answer to that question. So what we concluded then is that in some patients with POTS, there is a deposition or an increase of the phosphorylated alpha-synuclein. One of the points I didn't make is that in our hands, in the hands of 18 different groups across the world that have been studying phosphorylated alpha-synuclein for the past 20 years, the specificity of finding phosphorylated alpha-synuclein is essentially 100%. And what I mean by that is that no group has ever found a truly normal person to contain the phosphorylated alpha-synuclein. So we do think that finding is a highly pathologic finding in patients with whatever disease that they now have. Up until now, there has not been either a commercial test or an easy test that allow us to detect the presence of phosphorylated alpha-synuclein, but we can now do that in the context of a simple skin biopsy. And in that simple skin biopsy, we can also look at the epidermal nerve fiber density. So we get 2 really helpful pieces of information to try to identify those patients with neuropathic POTS. Do they have a neuropathy, a small fiber neuropathy? And do they have this abnormal protein that's being deposited? Now, one of the things that's very exciting about this work is that if you look in other situations with the synuclein diseases, we see that there are patients that have prodromes. So for example, if I get diagnosed with Parkinson's today, then over the previous 5 to 10 years, people that have that diagnosis of Parkinson's will turn out to have a number of autonomic symptoms. So constipation, gastroparesis, loss of smell, orthostasis. And everybody now is fairly convinced that that's what's called a prodromal phase. And that prodromal phase may take 10 or even 20 years to start to become symptomatic. So what that means then is that if we can detect the phosphorylated alpha-synuclein in that subset of patients that have it, it may allow us to be very early in the identification of these patients. And therefore, as we start to get drugs, and as I said, there's 34 different molecules now between phase 1 and phase 3 trials, it's going to be so much easier to prevent progression of these diseases than it's going to ever be to reverse them. So if you wait until someone has late stage Parkinson's, you're not going to fix that because so many neurons have been damaged. On the other hand, if you could identify them 10, 20, 30 years before that time, now you have a ton of room to really prevent these diseases from getting worse. Now, again, the very, very small study, retrospective study, not controlled study. And so we say that in science because what we want people to realize is this is not a definitive proof of any hypothesis. We do think the data shows an association between POTS patients with neuropathic features and the phosphorylated synuclein deposition. But again, it's not a longitudinal, blinded controlled study. We also do not know if these findings are an early stage towards the development of alpha-synucleinopathies. And as I said, there are 4. So pure autonomic failure, Parkinson's disease, dementia with Lewy bodies, and multiple system atrophy. So the point to make here is just because we identify it in people with POTS, and just because we know as people age, they develop Parkinson's disease and have the same protein, we don't know that there's a direct line between those two groups. Some people may just have alpha-synuclein and never progress. Some people may have it and progress, and we don't know the answer to that either. So we do want to caution either the excitement over knowing that we're now extending the prodromal phase, or the fear that if you find this, that this means that something absolutely means it's going to progress, because we absolutely do not know that. All right, just a couple of acknowledgements. So I did work with Chris Gibbons and Roy Freeman from Beth Israel as scientific advisors, although, as I said, all the patients and the work was done at Honor Health here in Arizona. Bailey Belair was our research engineer who did all the pathology, and just to thank all the patients who participated. And I can stop there and see if there's any questions.
Thank you so much, Dr. Levine. That was very informative. There's a lot of really great questions. And feel free to keep typing in questions here. I will try to get to as many as we can. So the first question is, do you see large fiber neuropathy in POTS patients?
So much less commonly, but you can. So the large fiber neuropathies are easy to identify. So you can do a physical exam and detect it, or you can do routine nerve conduction studies and detect it. So patients with diabetes certainly have neuropathy in POTS, but it's usually more that correlation where we think the diabetes is kind of the big elephant in the room, and then it's just causing multiple problems as a result of the diabetes. Likewise, the autoimmune conditions, right? So Sjogren's, I think 80% of people with Sjogren's have some form of neuropathy, either small or large fiber. And then Sjogren's probably does cause POTS as well. So, but it's probably more the Sjogren's just branching off and doing what it wants to do. But you can certainly see both. Yeah, it's not exclusionary.
Yeah. So there's a couple other sort of just regular neurology type question or neuropathy type questions. So this is a thought-provoking, good question. The person who asked this question is sort of thinking things through. Is it possible that people with neuropathy were simply not born with the missing nerves, or is it always acquired? They ended up they were born with the normal amount of nerves, and then later in life those nerves died off from something.
It's a great question. Yeah, all those are possible. So we like to think that, so if I lead a normal life until I'm 30 years old, and then something happens and I develop symptoms, we like to think that that's, that it began at the same time, so that you weren't born with too few nerves and then developed a problem. But that's not necessarily fair. So I will give you another example, which is, we now have the ability to do genetic testing at a rate we never could do before. And so I will see people who will tell me, a year ago, my neuropathy began. And I do genetic testing, and they have a clear genetic cause for their neuropathy, which means that they were born that way 30 years ago, let's say, but something occurred or they crossed some threshold and now they become symptomatic. So all of those are great questions. And I think like I think for those of us that do a lot of genetic testing, it's really stressed our understanding, because we find these people that are genetic that you never would have thought were genetic, so it's really interesting.
Yeah, I want to sort of echo that. A lot of people sort of divide up, there's genetic causes and autoimmune causes and diabetes. But the thing is that even autoimmune diseases and diabetes and other acquired conditions have a genetic basis at some level of depth, the more digging you do. So I think like everything in life, it's part nurture and part nature, right? It's your genetic substrate exposed to your environment and your life that you've lived.
Yeah.
So it's a great question for research that we certainly need a lot more of in POTS.
I'm sorry, I was just going to say one more thing. The other point to make about that too is that, when I say it's true, we only identify a cause of neuropathies 50% of the time. And I think the reason for that is that we really are stupid. So what I mean by that is I can identify one cause and one effect. But let's say you have a genetic disease, you're born with some genetic predisposition, and then you have a viral thing on top of that, and then something else on top of that. Now what's caused your neuropathy is 3 or 4 or 5 cumulative effects. Not in my lifetime will we ever be smart enough to figure that out. But I think that's why a lot of times we don't figure it out, because it's not just one thing, it's more than one thing.
Yeah. So speaking of that, we have a very smart patient scientist who's asked a question that maybe you can give our audience a little bit of background on the topic of the question that I'm sure you know. So the commenter asks, did Dr. Levine check these 22 patients for Fabry disease? Because she shares a journal article finding an association between Fabry disease genetics and deposition of alpha-synuclein in small fiber nerves. So maybe you could explain to the audience what Fabry's is first.
So, Fabry's disease is a genetic, inherited condition with a defect in an enzyme. And as a result of that defect in the enzyme, they develop a number of different medical conditions. Neuropathy is actually one of them, and can often be, despite heart, kidney, other things, oftentimes the neuropathy is one of the most limiting because it's very, very painful. And it selectively affects these small nerve fibers and the autonomic nerve fibers. So I wasn't aware of an association. I'll have to look at that with alpha-synuclein. I don't think I tested any of them, truthfully.
Yeah, this particular patient scientist, we've actually been discussing Fabry with some Fabry. There are some Fabry researchers in Europe who actually found that a very high percentage of their genetically confirmed Fabry patients met the EDS diagnostic criteria. So we're like, this is raising eyebrows. It can cause a dysautonomia. It might look like EDS.
Right.
And certainly something that, it's probably rare in our population, but there might be some undiagnosed Fabry in our community as well. So
the other important point about that is they now have an enzyme replacement therapy. So if you do make a diagnosis, you can get people onto therapy, which is, again, one of these, really exciting inherited conditions that we can now treat. So,
yeah. So here's a sort of similar question. Were there any autoimmune diseases identified in the group with the alpha-synuclein because we know that there is a known relationship between autoimmunity and subsequent development of Parkinson's disease?
Yeah, so we excluded anyone that had an identifiable cause. So that was so if they had Sjogren's, if they had lupus, if they had anything else, then we would not have included them in this study. But again, I think it's an important question. Sort of the hypothesis would be that the autoimmune group would not have it. But again, that could be a hypothesis that could be easily disproven.
Yeah. So I think I'm sure, I know you're planning future research on this, but just for everyone's benefit on this Zoom call, this is going to be extensively studied in the future. You don't have a finding like this that sort of just sort of disappears into a report on the shelf. I know that Dr. Levine and Dr. Freeman and Gibbons up at Harvard they're very interested in doing further research on this. And it's, Dysautonomy International has $100,000 or $200,000 research grants, but this is the type of research that takes millions of dollars to do very large cohort, long-term follow-up studies. So not only are we doing all the fundraising we can as an organization, we're also pressuring the government to put more funding into big POTS research that's expensive and really gets us the more in-depth answers that we need. So anyway, I know that you have big plans for future research. So I just wanted people to know.
We will do more. I promise we'll do more.
So here's a question that I asked by a lot of people. How can I, should I get tested for this, and how can I get tested for this?
Okay, so you saw my disclaimer, which is there's only one commercial lab that's doing this, and that's my lab. So take what I say with whatever grain of salt you like. I mean, the easy answer, obviously is talk with your doctor and see whether this is something that makes sense. I mean, to me, in my practice, I have begun doing it much more. But again, I'm doing it in the context of also understanding, not just looking for phosphorylated synuclein, because I'm hopeful that that number will be low, but also being able to look at the neuropathic POTS question by looking at the small nerve fibers. So if you're working with a physician that information would be helpful to, then I would say it's certainly a reasonable thing to do. Unfortunately, if we found phosphorylated synuclein in your nerves, as of today, there's no treatment for that. There's nothing. I mean, again, I think it's coming. I would think less than 5 years it's coming, but right now we can't. So especially neurologists, when we do tests, we always have to ask, so what? What would I do with the answer? So
right.
In this case, I think it's somewhat helpful because you learn, is there nerve damage, right? Is there phosphorylated synuclein? Is there epidermal nerve fiber damage? And that then allows you to know that you have neuropathic POTS. So I think it's pretty helpful. The how you'd get it done is it's done, as I said, by skin biopsy. We actually take 3 pieces. So one from your ankle, one from your knee, and then one from around your shoulder. But as I showed you, it's very, very small. There's no stitches. It just heals up like a scab. And it can be taken from anywhere and then sent to our lab.
Can you elaborate a little bit more on the cervical biopsy? So most of our dysautonomia population that knows about skin biopsies, which not everybody knows about, but most know about 3 leg biopsies, right?
Yeah.
Like ankle, knee, and thigh. So can you just explain a little bit why the cervical location is used in some conditions?
So there's 2 answers to that. One is we were wrong. So again, a hypothesis, right? So we do know that there are more nerves the closer you get to the center. So your ankle has the fewest, your knee has a little more, the hip a little more, the neck, shoulder area has even more. So we thought if you do this one, it would allow us to see more nerves and therefore see the phosphorylated synuclein inside of it at a higher rate. Now that we've done over 2,000 patients this way, we actually ran the numbers and it turns out they're equally distributed. So there's nothing magic about that. So it was our hypothesis, but because we've established all the controls that way, we try to stick to it. We know that if you do 1 biopsy, we can detect it 75% of the time, 2 biopsies about 85, and 3 biopsies about 95. So it's just a volume business. The more nerves you see, the more likely we are to see it. But I did a very nice young woman on Friday with POTS, and I did her ankle and her knee. She was like, "You're not doing my neck, I'm done." I'm like, "Okay, so we'll look at 2, fine."
Yeah.
So there's nothing critical, truthfully. Yeah.
Can you, sort of on that note, can you explain the difference between a length dependent small fiber neuropathy and a non-length dependent small fiber neuropathy?
So this is something I've published a lot on in the last 5 years. And others, but so again, if you think about most diseases that affect nerves, diabetes, B12, chemotherapy, alcohol, HIV, you name it, they almost all damage the longest nerves first. And so the nerves start in your spinal cord and they go out to your arms and legs. So the longest nerves that anyone has are the nerves that go to your toes. And that is why the book that was written was called Numb Toes, Burning Feet, because almost all neuropathies start in your feet. That is called length dependent. There are some people that don't have that. There are some people whose neuropathy is in their chest or their back or their head or their shoulders. Yeah. And that is called non-length dependent. And you can define that clinically just because the person says my burning's on my chest, not my feet, or you can define it based on the biopsies. And that's the reason that we recommend 3 when you're doing the biopsy for small fiber to be hip, knee, ankle, because then you compare how's the damage at the hip to how's the damage at the ankle. And the importance there is that non-length dependent have a much, much higher association with autoimmune diseases. So when you think about all the weird antibody syndromes like TSHDS and FGFR3 and Sjogren's and lupus and sarcoid, those are not always, but they're much, much more likely to be non-length dependent. Whereas the other diseases like diabetes and B12 are really never non-length dependent.
So we have a bunch of questions about if you had a previous punch biopsy at Corinthian or another lab, can you get it rerun for alpha-synuclein?
Well, again, since I work at both Corinthian and C&D, the answer to that one is probably yes. So typically when you have the biopsy done, we keep the leftover tissue for a couple of years. Now that assumes there was enough tissue and so forth, so no guarantees, but as long as there's enough leftover tissue, we can ship it because they're actually, the labs are in different states. Corinthian is in Dallas, C&D is in Arizona, but that can be done, and theoretically that could be done from other labs as well, as long as they save the tissue.
Yeah.
Yeah.
Does insurance cover the alpha-synuclein testing in a younger POTS people?
So pretty well. It covers it as well as it covers the small fiber testing.
Okay.
Okay. So for older people, so the vast majority of people that we do the synuclein testing on for Parkinson's and dementias, they're Medicare age, and Medicare pays for this very well. So that's kind of a no-brainer. When it comes to the commercial plans, it's always a little more challenging, but we've had pretty good success with most of the major carriers in doing it. Yeah.
So speaking of age, if I'm remembering correctly, the youngest patient in your study who had the synuclein was 22.
Yes.
Is that right? That was like a month ago that I read that.
Yeah.
but it's important because usually this kind of pathology finding is not usually studied in young people, right? They're really, I don't even know how many studies are there out there where we're screening 20-year-olds for this. So I think it's oh, actually, this is a related question. There were sort of 2 or 3 questions about age. So I think it's important for people to understand that it's not just 60, 70-year-old patients who have this. It's
Right.
sometimes in young people. But a good question was what type of onset these patients had, because we know that in POTS, some people have a post-viral onset, some people have a post-concussive onset, some people have just a more insidious onset where there's no definable time that they got sick.
Yeah.
And do you know the age or the type of onset any of these patients had?
So I don't believe there were any that were post-concussive in the group. And I don't know the percentages, but I do know that some of them had the, what I think is not a typical acute presentation, where there's almost a defined week or month when something begins. Right. And then some had the insidious kind of onset. There were a couple of the ones in their 20s and 30s where it began as a teen. So some of them did start younger, but I don't see under 18. So that was about, I think that's about right.
Did any of the study subjects that had the alpha-synuclein have Ehlers-Danlos syndrome?
Yes.
Yes.
Yeah, I'd say, don't quote me, but it's probably about a quarter of them.
Right, which is pretty similar to the overall rate in POTS.
Yeah.
There's different studies that say different percentages, but most studies fall around 25 to 33% of POTS patients having EDS, hypermobile EDS most often. So that's interesting. We did get some questions like, from patients saying, well, if I have an autoimmune disease and EDS, does that rule out the possibility that I have this alpha-synuclein problem? And it seems like the answer is probably no, but I'll let you answer that.
Yeah, no, you're right. So, well, we didn't study that group. So if you had one or two things that could
Right.
be considered causes, we sort of excluded that group because we were really kind of focusing on the idiopathic neuropathic group.
Right.
But so I don't know the answer. I would guess that we would probably still see it in some. Again, would be my hypothesis to be disproven. But I think until we do the study, we just don't really know.
Or there's a question about do people of different racial and ethnic backgrounds have different rates of POTS? And same question, different rates of alpha-synuclein problems?
I'll let Lauren answer the first one because I don't know the answer.
Yeah. Well, so on the first one, we know that who's getting diagnosed with POTS right now is
Right.
very, very largely Caucasian people.
Yeah.
But that doesn't mean that we're actually accurately, we know all of us who've lived with this condition and our family members know that we have a really hard time getting diagnosed, right? We often wait years and have to see lots of different doctors. We're told it's all in our head. We kind of get blown off. So I would say very cautiously that it might be Caucasian predominant, but we're probably missing a lot of people of different racial and ethnic backgrounds. And the reason I know that is because we are seeing research coming out of Japan and China and South Korea and India, and they have large populations of POTS patients in their studies and their centers. And yet in the US, the vast majority of our research, it's like 90 to 95% Caucasian. So we're obviously missing those patients. And so there are lots of very complex societal problems and socioeconomic factors that prevent people from getting accurately diagnosed. So that's a very long way of saying it might be Caucasian predominant, but I wouldn't trust our research data enough to say that definitively. At least we know that's who's getting diagnosed in the studies right now, but we probably have a lot of work to do as a physician and patient community to reach a broader audience.
Yeah.
So no, same question on alpha-synuclein, which I don't know the answer to.
So that one's easier actually, because we do know the answer. So 100 years ago, a neuropathologist that was studying Parkinson's disease patients discovered these blobs. They didn't know they were proteins, but he discovered these blobs in their brains, and they were called Lewy bodies because that was his last name, Frederick Lewy. And then 40 years ago, people discovered that what was inside the Lewy body was synuclein. So it's been studied for really almost 100 years now. And so everyone with Parkinson's disease, regardless of race, age, whatever, has alpha-synuclein and the phosphorylated alpha-synuclein within their brains. And the same with dementia with Lewy bodies, and the same with pure autonomic failure. So we know that that finding is universal, and that having phosphorylated synuclein in those patients is universally true. Obviously, this is the only data in POTS. And as Lauren said before, I'm pretty sure this paper is the youngest description of people having phosphorylated synuclein within their nerves. And I'm now actually also, in addition to all the things that Lauren said I want to study, which I do, I also really want to get my hands on some kids. Because
that doesn't sound good.
Exactly. That's the point. Not quite that way.
I know what you mean.
I want to get some skin from kids. Probably sounds worse. Okay.
Some biopsies. You want to take chunks of flesh from children.
Exactly.
And this emphasizes the importance of the adult and pediatric researchers in the POTS world working together, which right now they really don't. They live in these two separate spheres. And so that the idea of having multi-center collaborative research is a huge priority for us. So it's good to hear that. Let me ask you a couple more questions because I don't want to keep you all night, but so let's see. So is there any consensus that POTS may be an early neurodegenerative disease? I think this is so new. The answer is probably no, but what have your colleagues thought about this?
Well, so there definitely isn't a consensus. I would say that for sure. But I think there's been a belief that, so there's a disease called pure autonomic failure where patients really have terrible dysautonomia. We know that there's a condition called neurogenic orthostatic hypotension, which is clearly where they have failure of their autonomic nervous system, but not as bad as this. And so the question becomes, is there a continuous stream that some patients follow? We clearly don't think all patients do this with POTS. It's probably not even the majority of patients that do this with POTS, but there may be some patients where POTS is the beginning of something, which 10 or 20 years later could be neurogenic orthostatic hypotension. And then another 10 or 20 years later could turn into an autonomic disorder. So I think, Roy Freeman and Chris Gibbons probably would say that makes sense. But again, that could be 1%. It could be 20%. We don't know. And we're not even really sure that finding the phosphorylated synuclein is the predictor of who those people are going to be. But we've all seen, Roy, Chris, and I, all of us that do this work, we've all seen some POTS patients that it just goes badly for, right? They just do worse than others. Yeah. And so we do think that there's something in there. Again, multiple different types of POTS, multiple different prognoses. And it would be interesting if this finding, sort of says maybe there's a little worse prognosis, but we just don't know that yet.
Right. I just want to share, like, on a personal note for patients who, hear this information, and it's scary, right? You're young, and, you were told by so many doctors that you're going to get better. And, I'd say the vast majority of POTS patients do improve to some degree over time when they find a good doctor and they get the right treatments. I'm not saying it's an easy journey or a magical cure, but most patients do get some improvement. So what's really scary is if you have this problem, it sort of leaves more of a doubt, like, am I going to ever get better? Like, am I going to get worse? And so me personally, I actually have a strong family history of Parkinson's disease. So even before this research came out, I was thinking, am I going to end up with that someday? And I have Sjogren's syndrome, which increases your risk of developing Parkinson's disease later in life. So even knowing all that, I'm not worrying about this on a day-to-day basis, because I know, unlike POTS, Parkinson's gets a ton of research funding. And so there's a lot of really, really incredible research happening in the Parkinson's space. And I am pretty confident that by the time I'm old enough to have potential Parkinson's, that there's going to be, really great treatments. And I'm just telling you guys this because I don't want people being terrified to find out what's going to happen to me 60 years from now. You can't predict what's going to happen with your own body or with science. And so I think that's why it's so important that we keep pushing for the research to get, better treatments for all the different types of dysautonomia, not just POTS, not just autoimmune POTS, not just EDS POTS, like big picture. So do you have any final thoughts you want to share with us? I don't want to keep you all night.
Yeah. No. I think that was great. Yeah, I just sort of echo what Lauren said, which is this is really 100 years of research into this one protein. And I'm really, really optimistic that for the patients with Parkinson's and dementia with Lewy bodies and those significant diseases, that we're going to have something that really treats this within the next 5 years. And even if you do get the test, and even if you do have, phosphorylated synuclein, it's telling you that there may be something you have to worry about 40 years from now. So as Lauren said, I wouldn't spend every day worrying about it, but if you knew, you would pay attention to when maybe the trials come around to be involved in, or
Yeah.
pay attention to when the drug comes out, that you could be one of the first to get it because you know that you have it, and then be excited about the fact that you'd be treating this decades before it becomes a bad disease, like Parkinson's or dementia with Lewy bodies, and really prevent it. So I think there's a lot of optimism. I think hopefully this will help us understand POTS better, and really be part of a bigger treatment for a lot of significant diseases. So.
Yeah, just if anyone's looking for a little info on alpha-synuclein research, I would encourage you to check out the Michael J. Fox Foundation for Parkinson's Research. They have really great stuff. And little side note, I actually met their CEO at a conference when we first started Dysautonomia International, and they lent us some of their grant application materials and kind of other like starter paperwork for a health nonprofit. And they said to me, "Did you know that 67% of Parkinson's patients have dysautonomia?" And I said, "Yes, I did. It's on our website." But they're a much bigger organization with a ton of really successful research grant program that we are trying to emulate on a smaller scale. And we don't have a Michael J. Fox, but eventually, I think we might find that person. So thank you so much for your time. Thanks for everyone for tuning in. This is being recorded and it will be available to everybody who registered for the event in about a week. Dr. Levine, we've had a few people ask, would you share some or all of these slides?
Yeah, do you have them? You probably don't have my.
I do. I always ask before we share them.
Yep, absolutely. Yep.
Great. So we'll share those when the video goes up. We'll put a link to the slides too. So thank you so much for your time and expertise.
Thanks, you guys.
And
yep.
Thanks to everybody for tuning in. Have a good night.